Culturing is our way of growing organisms for study. We begin with an inoculum or a sample of culture. It is grown on a medium or collection of nutrients that allows growth of the organisms. The culture is the group of microorganisms that grow from the inoculum. Sometimes the medium is solid, other times it is liquid in the form of broth. Clinical specimens are samples of human material examined for the presence of microbes. When studying microbes, we need to look at a pure culture composed of cells arising from a single progenitor. They will form CFUs, or colony-forming units, where the single cell or group of related cells form the colony. To create a pure culture, we must use sterile equipment that is free of microbial contaminants. The organisms must be incubated or given time to grow. One of the most common ways to achieve pure cultures is to use streak plates. A sterile inoculating loop is used to spread the inoculum over the medium to separate the organisms, to isolate the colony-forming units. Pour plates are another technique where colony-forming units are separated by using a series of dilutions that are mixed and poured into petri dishes. Other times, with large organisms, they may be picked up by micropipettes. The composition of the media can vary. In defined media, the exact chemical composition is known. This is actually quite tedious and expensive, so it is not used all that often. Complex media contains nutrients released by the partial digestion of something like yeast, beef, soy, or another protein. Selective media may be used to favor the growth of some organisms while inhibiting the growth of others. All of these media can generally be placed in petri plates or in slants where the agar solidifies at an angle, allowing a larger growing surface while the bottom is almost anaerobic. A differential media may be used to create a visible change in the media or colonies to differentiate the types of bacteria growing. The selective and differential media are both very useful in helping to isolate a specific organism. A stab culture may be used to inoculate the anaerobic portion of the media. Reducing media will contain compounds like sodium thioglycolate that combines with the oxygen to make it anaerobic. Petri plates can also be placed in a sealed container with a reducing agent. Clinical specimens are carried in transport media, which is designed to maintain the ratios of organisms, particularly in stool. Some organisms require living cells in animals or cell cultures. Rabbit and bird eggs are commonly used. For cultures needing low oxygen, the carbon dioxide incubator may be used to mimic the environment of the gastrointestinal tract. This can be done using a gas pack or a candle jar. Candle jars are cheaper and very easy to use. Caphophiles are organisms which grow best with relatively high carbon dioxide and low oxygen levels. An example is Neisseria gonorrhea. The enrichment cultures used a selective media designed to increase a small number of chosen microbes to an observable level. Cold enrichment cultures are used to enrich a culture with cold-tolerant species such as the Vibrio cholerae. Those are incubated in a refrigerator instead of at 37 degrees Celsius. There are a few ways to preserve cultures. Refrigeration is best for short periods of time. Deep freezing to -50 degrees to -95 degrees Celsius will allow organisms to be stored for years and then restored. With lyophilization, the organisms are freeze-dried in a vacuum to remove the water. They can last for decades and then be revived by adding them to liquid culture media. This is how organisms are ordered for use in the lab. When bacterial populations grow and divide with binary fission, a cell divides to produce two equal-sized daughter cells. It begins by replicating the chromosome and then elongates and pushes the chromosome apart. A new membrane and a wall will form in the middle. When the septum is complete, the daughter cells can remain attached or separate depending on the species, and the process itself repeats. This logarithmic or exponential growth is much faster than arithmetic growth. One cell divides to make 2, then 4, then 8, and 16, and so on. The time it takes for a bacterial cell to grow and divide is referred to as the generation time, and this varies. We have different phases of microbial growth, which can be plotted in a growth curve to show the number of organisms growing in a population over time. During the lag phase, the cells are adjusting to their new environment and do not reproduce immediately. This can range from less than an hour to days. In the log phase, the population increases logarithmically. During this phase of rapid growth and reproduction, cells are more susceptible to antibiotics because they are taking in more nutrients and are more likely to take up the antibiotic. It is also the best time for staining. During the stationary phase, the number of cells dying is equal to the number of cells being produced, and the population numbers stay the same. The metabolic rate of survivors decreases, so fewer cells are produced. A chemostat may be used to maintain the population by removing waste and adding fresh medium to postpone the stationary phase. In the death phase, nutrients are not added and the waste is not removed, so more cells die than are produced. However, some cells may remain alive, especially if they produce endospores, so one should not assume all the organisms are dead. Sometimes we need to count organisms, and this may be done directly or indirectly. One method of directly counting organisms is the viable plate count. Here the sample of organisms is diluted through serial dilutions and then plated in petri dishes and incubated. After incubation, the colonies on the plates are counted and the dilution factored in to estimate the number of bacteria per milliliter in the original culture. It is best to count plates with approximately 25 to 250 CFUs to be sure there is adequate growth, but not too much to be able to accurately count. Membrane filtration may be used to directly count bacteria. This is used for very dilute samples in a rather large volume of fluid, such as water samples. The sample is poured through a membrane and then transferred onto a solid medium. The number of colonies is equal to the CFUs in the original large sample. Another option is to do a microscopic count if the organisms are more concentrated. A small sample is placed in the counter, which is a special glass slide with a grid. The number of organisms is counted in several squares and then used to estimate the number of bacteria per milliliter. We will be doing several of these techniques in the lab. There is also a link here to read on membrane filtration on your own. Cells can be counted electronically using a Coulter counter, which counts cells as they interrupt the electric current flowing across a narrow tube. Flow cytometry uses light transmission to count cells. There is also a video here that you can watch on your own time that gives plenty of information on flow cytometry. In a situation where it would be necessary to determine the number of bacteria you have frequently, these electronic or light techniques would be used over culturing and doing serial dilutions just because the results are so much quicker. The MPN, or most probable number, is a statistical estimate. It is based on the more bacteria in the sample, the more dilutions will be required to reduce the number of colony-forming units to 0. Indirect methods may be used, such as a measure of metabolic activity. Here, growth is inferred from the rate at which the population utilizes nutrients and produces waste. The dry weight of the organisms, which are dried, filtered, and weighed, may be used to estimate their quantity. Spectrophotometry may be used to measure turbidity or light transmission. As bacteria reproduce, the culture becomes more cloudy or turbid. The measurements are compared with a scale to approximate growth. The technique used depends on the lab's need for accuracy, how frequently the numbers are needed, and how quickly they need the results, and the availability of the equipment, which is often very expensive.